This is the first of a series of videos that should help you through Chapter 11 and learning how to do the various different kinds of genetics problems that are included in that chapter. Okay, so first of all, you learned in the video that we watched, the Cracking the Code, about Gregor Mendel and his history. We know that this was taking place long before they even saw chromosomes in cells, much less knew that DNA was the genetic material. That was 100 years later. So Mendel was really doing these discoveries without really having any kind of a visual. So it was kind of a black box that he was figuring out the patterns of inheritance. Nevertheless, he was able to use patterns to come up with the basic principles for how traits are passed from one generation to the next. All right, so first of all, his ideas, one of the things he came up with was that alleles account for variations on characteristics. He never used that term alleles, but he called them hereditary units. And these hereditary units are the things that account for variations between individuals. He also was able to figure out via his patterns that each individual must have two of these hereditary units or alleles, which they inherit one from each parent. We now can see that on chromosomes, and you guys have already learned that cells are diploid, and so you understand that there's pairs of chromosomes, but Gregor Mendel didn't know any of that. All right, and then he also was able to figure out that if these two alleles are the same, that you could have a particular trait that's related to those alleles. But if the two alleles are different, then one of those alleles may be the dominant allele, and that's the one that you can see, the visual outcome of it, whereas the recessive allele is the one that has no effect on the outlook or the phenotype, we say, of the organism. He also then was able to say that if you have these two units, only one of the two of them can be passed in a sperm cell or an egg cell to an offspring. So it's called his law of segregation. Without even being able to see mitosis or meiosis, he was able to see that these, in meiosis, that these two hereditary units end up in separate daughter cells. All right, there are other common terms related to this chapter that we haven't covered, but they should be things that should be familiar from regular biology. Homozygous versus heterozygous, so we can break this down into three basic collections of genes that an individual may have. An individual may be homozygous dominant, homozygous recessive, or heterozygous. Now, homo is a prefix that means same. So if an individual is homozygous dominant, that means that they have two dominant alleles. We would write this, if you had to write down or document this, we write this with capital letters. So an individual that is homozygous dominant would be big A, big A. Homozygous recessive, on the other hand, once again, same, but both of the alleles are recessive, so you use lowercase letters for that. Word to the wise, try to pick, if you can, letters that you can distinctly tell the difference between uppercase and lowercase. You may want to really exaggerate these differences in sizes of these letters so that your handwriting doesn't screw you up. One thing that I do is, let's say I had to use S's that don't have a big difference between the uppercase and the lowercase. I write the uppercase just normal, and then I write the lowercase as cursive so that I can keep track of which trait is which. Okay, now back to the different kinds of homozygous versus heterozygous, the different kinds of, we call them genotypes or gene collections that an individual can have. They could also be heterozygous. Hetero means different, so an individual that's heterozygous has one dominant allele and one recessive allele. All right, another term, genotype versus phenotype. The phenotype is the physical outward expression of the gene, so it's the result of having a particular collection of genes, whereas the genotype is what we just described up above, homozygous versus heterozygous. That's the collection of genes that an individual actually has as the genotype. So if we take a look at this diagram here, you can discern between those two different terms. Notice that phenotype, those are physical descriptions that the individual may have, purple flowers versus white flowers. Whereas genotype, that's telling me the gene collection that the individual may have, being homozygous, heterozygous, homozygous recessive, all of those things are genotypes. Okay, so now let's go through a basic genetics problem. Many of the problems we're going to be doing are going to be a step above this type of a problem, but if you don't have your basics down, then you're going to have difficulty building on this general core idea. All right, so the steps are a lot like dealing with a word problem in math class. You got to read the problem and pull out the important information. Now, if possible, if the parents' genotypes are indicated or alluded to in the question, you want to write those down. I still write those down after so many years of doing genetics problems. I still do that. Punnett squares may be used. You may get to a point where you don't need them anymore, and I'm going to emphasize using the laws of probability instead of using Punnett squares at times. But it is a tool that I want you to know how to use, and so I'm going to start out with it in the basics. And then finally, make sure that you answer the question as it is being asked. Sometimes you're asked for ratios, sometimes you're asked for percentages, sometimes you're asked for chances. Make sure you respond to the question appropriately. Okay, so let's try a question. Here is a basic genetics problem. Please read through this and see if you can use your skills of using a Punnett square to go through and solve this. So pause the video, come back, and check your answers. Okay, so taking a look at this problem, the first thing that I did is I did go through and I wrote down the genotypes of the parents indicated in the problem. One parent is heterozygous and the other parent has the recessive phenotype, so it must be little e, little e. And I set up a Punnett square. A Punnett square is a tool that's just going to tell you the possible combinations from a set of parents, and it also can be used then to figure out the probabilities of each one of those combinations. So you take one parent and you put their alleles. Remember, they can only give one of the two, so you have to separate them. So one parent's alleles go there and the other parent's alleles go there. And then you put them together in the boxes, and that shows the combinations that could be found in a particular offspring. So this question then is asking for what is the chance that their first child will have free hanging earlobes, which is the dominant condition. So of the 4 boxes, 2 of them are showing the dominant condition, so it's 2 out of 4, or you could simplify that to a 1/2 chance. Another way that you could give a chance is as a percentage. So a percentage would be an acceptable answer for this one. So saying a 50% chance of the child having free hanging earlobes is also an acceptable answer. All right, so I'm going to go through test cross here just because there's not that much beyond a general problem. The only thing is that a test cross is wondering about if you have an individual that has the dominant phenotype, purple flowers. You don't know whether that individual is big P or big P little p. So let's say you really needed to know for one reason or another. For breeding purposes, you needed to know the genotype of that individual. A test cross will cross that individual with an individual that has the recessive phenotype, because then that has a known genotype, so you can tell from the outcomes. After you do that cross, if this individual is, let's say, heterozygous, then you would see in half of the offspring, because the purple individual is heterozygous, it's carrying the recessive white flower color allele, you would see then that half of their offspring would have white flowers. On the other hand, if that purple individual is homozygous dominant, then all they have are dominant alleles. Therefore, they're going to have children, offspring, not really children, offspring plants, that all have purple flowers, and they all would be heterozygous from this cross, all getting a recessive allele from the white parent and a dominant allele from the purple parent. So that's called a test cross. That would only be done in certain instances where you are wondering what the genotype of an individual is. All right, so that's the end of the first video. In the next video, we will go through some next steps in genetics problems.